Hello, everyone, and welcome to GenScript's 2020 Fall Webinar Series. My name is Grace Tan, and I am the Product Manager for Reagent Antibody Services at GenScript. I have the pleasure of moderating today's webinar. If you have a question for the speaker or the organizers, please type it in the chat box for us to see. The speaker will be taking your questions at the end of the webinar. However, if we run out of time and she is unable to get to your question, we will be sure to send her a response to you via email. So without further ado, let me introduce our speaker today, Professor Joe Morris. Dr. Joe Morris is a professor of molecular genetics within the Institute of Cancer and Genomic Sciences at the University of Birmingham in the UK. Her research interests span post-translation systems and how they relate to the regulation of mammalian DNA repair and replication. Her recent work relates to how the removal of SUMO is critical for the correct timing of events in DNA repair, and how the addition and removal of ubiquitin by BRCA1 and USP48, respectively, control 53BP1 placement and resection. Today, Professor Morris will discuss her recent work, which reveals how the regulation of BRCA1 in replication fork protection differs from that in promoting recombination in homologous recombination, and how that in turn may have implications about the role of fork protection in cancer predisposition. Thank you, Joe, for joining us today. Please take it away.
Thank you, Grace. Hello, everybody. So let me just start by saying, yes, I'm from the University of Birmingham in the UK. Usually a very beautiful campus, under slightly different circumstances at the moment, but still beautiful in the fall. And before I start, I always like to say who's done the work. So in my group, that was Manuel Martin here, who was a grad student when he did this work, along with postdoc Ruth Densham, and our collaborators, Jiadong Zhang at Imperial College and her MRes student, Stephanie. So today, and this group of people helped an awful lot, I forgot to mention. And today we're going to be talking about fork protection, by which I mean the stabilization of replicated DNA that comes across a secondary structure or a problem and is forced to reverse into what's often called a chicken foot structure like this, and that somehow protects this recently replicated region from being digested by nucleases such as DNA2 and mRNA11. It was described by Catalina Schlacker when she was in Maria Jason's lab a few years ago. So I'm very interested in BRCA1. This is a classic BRCA1 pedigree here, where there are individuals with very early onset breast and ovarian cancer. And we know, of course, in the population that individuals with a BRCA1 mutation are prone to breast cancer at a very early age. And we tend to think of BRCA1 as important in homologous recombination and not necessarily in replication forks. In homologous recombination, it has three roles. The first is in the counteracting of 53BP1, RIF1 shielding, and the fill-in that's promoted by POL-alpha and CST that allows resection to happen, a little chewing back of the ends here. The second role is in promoting PALB2, BRCA2 loading of RAD51 onto those exposed 3' single-stranded DNA ends. And the third, recently described one, is in stabilizing the synapse here in the D loop. But in replication fork protection, we're not really sure what it's doing. And that was the question that we wanted to address. How is it that BRCA1, along with actually a number of other genes involved in homologous recombination, seem to be critical for this function? This function of protection is mediated by RAD51, so it's perhaps not surprising that many of the proteins involved in homologous recombination are also important here. So this is BRCA1, a fairly large protein, and we wanted to simply start by asking which portions of this protein are required for replication fork protection. We steer clear of the BRCTs. We know they're important for BRCA1 recruitment, but we started over here looking at the N-terminus and the ring domain, because we know here's the ring coordinating around two zinc ions. This interacts with another protein called bardon, and together these form an E3 ubiquitin ligase. We recently showed that a small amino acid change on bardon on the partner can interrupt the ability of BRCA1 bardon to function as an E3 ubiquitin ligase by affecting the ability to bind to the ubiquitin portion on the E2 loaded with a ubiquitin, such that the mutant is much slower at transferring ubiquitin from the E2 to lysines of substrate proteins. This is a really helpful mutation because it allowed us to separate this specific function of ligase from many other functions that BRCA1 has. So it disrupted the ligase function, but didn't have much impact on dimerization, stability, or localization into foci. We know that this is important for the placement of 53BP1. So in cells expressing this mutant form of BRCA1 that's poor at acting as an E3 ubiquitin ligase, we get the same placement of 53BP1 here shown in orange as BRCA1 within these DNA repair foci detected by confocal microscopy. So we know that the E3 ubiquitin ligase function of BRCA1 is important for positioning 53BP1 and in turn for allowing resection. I'm not going to go into it in great detail, but we know that this E2 ubiquitin interaction is important for putting ubiquitin onto H2A. It's the target. Many people have shown that H2A is the target, and we agree with that. And that in turn, that recruits a chromatin remodeler called SMARCA-D1, which is responsible for the movement of chromatin and the movement of 53BP1. This activity is required for resection. It's required for olaparib resistance, camptothecin resistance, but not required for hydroxyurea resistance or acetylcholine resistance. And that's why I'm telling you about this, because I think it's going to be important for the next part of my talk. So, is the E3 ligase activity important for this replication for protection function? Now we measure this using nucleotide analogues. So cells are incubated with two different coloured nucleotide analogues before being incubated with hydroxyurea, which causes replication forks stalling. Under these conditions, they don't collapse, they simply just stall. So if a fork is stable and it's not digested, the two colours are represented to the same degree. They have a ratio of 1. If a fork is not protected and the nucleases can get at it, then obviously that ratio is going to be very different because the second portion of that nucleotide analog will be digested back and ratio is less than 1. So, for example, here we have cells treated with a non-targeting siRNA. They have a ratio of near 1 of the two nucleotide analogs. But when we deplete BARD1, or if we were to deplete BRCA1, we would see a much shorter fork, ratios near a half. We put back wild-type BARD1 that's siRNA resistant, we get a rescue. But here's the interesting thing. What happens when we put back this mutant, which is specifically deficient in E3 ubiquitin ligase activity? Surprisingly, they're fine. So there's no defect associated with the E3 ubiquitin ligase function, despite the fact this is super important for resection. So this isn't important for replication for protection. The next thing we looked at was the coil-coil, because that's important for interaction with PALB2, BRCA2 for loading RAD51. And I told you that RAD51 is critical for the protection of replication forks. So we made a mutation, here it is in the coil-coil, and we know that cells complemented with this form of BRCA1 are very sensitive to cisplatin, as expected. However, remarkably, this mutant is completely capable of rescuing fork protection, and the forks are protected fully. So that really surprised us. We know that BRCA1, BRCA2 are important for replication fork protection. We did expect that this interaction would be critical. So to make sure that that's right, we then looked at a deleted form of PALB2 where we took the N-terminus off, removing the coil-coil required for interaction with BRCA1. Again, cells complemented with this form of PALB2 are very sensitive to cisplatin, and again, this mutation can completely rescue fork protection. So we're fairly sure that the canonical PALB2 BRCA1 interaction is not required for fork protection, even though PALB2 itself is. So that left us with a quandary. How is BRCA1 functioning in replication fork protection, since it neither needs the activity responsible for promoting resection, nor the activity responsible for promoting PALB2, BRCA2, and RAD51 in homologous recombination? So whilst we were intrigued by this, we had been working on a phosphorylation site here just after the BRCA1 ring region. And together with GenScript, we generated an antibody, or rather GenScript generated an antibody using a peptide of phosphorylated BRCA1 phosphorylated around this serine here. And this was a really useful tool. We could use it in vitro, so we generated BRCA1 BARD1 and incubated it with various CDK forms, and found that CDK2 in particular was capable of phosphorylating BRCA1 at this point. And if we mutate this serine here, we don't see any signal with this reagent. In cells, similarly, when we mutate the site, we don't see any interaction of the phosphoantibody with BRCA1. And if we inhibit CDKs, we see a reduction in the signal. So it looks like we've got a CDK-regulated phosphorylation site just upstream of the BRCA1 ring. Then we'd ask the question, well, where is this phosphorylated form of BRCA1? Might it be related to replication? To ask that question, we use this form of proximity-linked ligation, in which we label nascent DNA, the recently made DNA, with EDU, bind that to biotin, and then use antibodies to the biotin and use our phospho-BRCA1 antibody to ask whether phosphorylated form of BRCA1 might be close to nascent DNA. And this is what we get, these rather lovely little foci that are increased when we treat cells with a replication stalling agent, hydroxyurea. And if we quantify these, we can see it goes up in response to hydroxyurea, but that increase is not seen when we deplete cells of BRCA1, when we treat cells with a phosphatase, or we treat cells with an inhibitor of CDKs. So it looks like indeed we see phosphorylated BRCA1 at sites of stalled replication. So is this important for replication fork protection? The answer, of course, is yes. So this single point mutation was capable of resulting in cells that had poor ability to protect forks from degradation. But intriguingly, a phosphomimetic put at this site can rescue. So it looks like we've found something that is important for replication fork protection. And this made us think about that third role of BRCA1 in homologous recombination. This interaction that BARD1 has with RAD51 in synapse stability. So this was described by Patrick Sung's group a couple of years ago now, and they identified this site in BARD1 that prevents interaction with RAD51, which is important for the third function of BRCA1 homologous combination, not the resection and not the promotion of BRCA2, but contacting RAD51 directly and promoting synapse stability. Might this also be important for fork protection? So we made the same mutation as described in Patrick Sung's paper, and indeed, yes, it is. So here we go. This is the mutation here, and it cannot restore fork protection. So here we go. We've got this intriguing finding of a phosphorylation site on BRCA1 and a RAD51 binding site on BARD1, both required for replication fork protection. Now, we don't know what the structure of this region is. It's just upstream of the NMR structure that Rachel Kevitt described, but we might suppose that they could be close. Perhaps this is in some way related to the RAD51 binding. We also noted that this SP site here is a potential PIN1 interaction site. And we know from work of David Cortez that PIN1 is indeed enriched at nascent DNA that's treated with hydroxyurea. So we asked the question, well, perhaps PIN1 is also required for replication fork protection. And indeed, that's what we find. So when we deplete PIN1, we get short forks, or we deplete PIN1 together with BRCA1, we don't get anything shorter. So it looks like this might be in the same pathway as BRCA1. So that really intrigued us. So what is PIN1? So PIN1 is a phosphospecific peptidyl prolar isomerase. And what that means is it's capable of moving the backbone around proline. So you probably remember from chemistry classes that most, in fact, all of our amino acids, with the exception of PIN1, are in trans. So the backbone goes down here like this. The exception is proline because it's got this 5-membered ring, and you can imagine this can be swapped around in either direction. So it can be either in cis like this or in trans. And that's the role of PIN1. It can catalyze the movement of that backbone from cis to trans and back again, although one direction is more favored. It does it in a specific phospho binding way as a WW domain, which only interacts with phosphoserine and threonines preceding the proline. So might this be an important feature of the RAD51 interaction. So to answer that, the first thing we did was we made BRCA1 BARD1 in vitro, and we made a serine to a mimetic for the phosphorylated state and incubated it with and without PIN1 and RAD51. And what you can see from this plot is that only the phosphomimetic incubated with PIN1 gave us a decent amount of RAD51 binding. Actually, the wild type didn't, even when it was incubated with PIN1, suggesting that perhaps indeed PIN1 is somehow able to regulate access of RAD51 to this BARD1 RAD51 binding site. To take that a little further, we mutated the proline itself. As I said, because all amino acids with the exception of proline are in trans, we can at least ask the question, what happens if this residue is in trans? So here we mutate it to an alanine to hold it in this translocation, whereas of course the wild type can be in either. We do that in vitro using the P115A form of BRCA1 in a BRCA1 BARD1 heterodimer, and only the mutant form is capable of bringing down significant amounts of RAD51. You can see it's quite a lot of RAD51, suggesting that indeed if we hold this in a more trans-favoured form, the ability to interact between RAD51 and BARD1 is increased. To do this in a more physiological way, Stephanie Tai in Xiaodong's lab made full-length BRCA1 BARD1 with and without this mutant in it, and asked the question, how can trypsin digest these proteins? And what she found was that it didn't matter whether BRCA1 had a P115A or not in terms of its access to trypsin. Trypsin was able to digest it at the same rate regardless of the mutation. But that was very different for BARD1. So in the presence of P115A BRCA1, BARD1 was more accessible to the trypsin and was digested slightly faster, suggesting that indeed this change at P115A affects the ability to access BARD1. So we can do the same trick in cells. The wild type, of course, can go in either conformation. The serine to alanine mutation is presumably in the conformation it was in before it was phosphorylated. The P to A form is going to be in a trans-favoured conformation, and we can do a double mutation that's in a trans-favoured conformation, but it also cannot be phosphorylated. And that can ask the question, which is more important, the phosphorylation event or the isomerization event? And here's the data. So what you see, as before, the serine to alanine mutation has short forks, they're not protected, but importantly, the double mutants, the P to A on the same background rescues, showing us that the trans form is able to rescue the need for the serine to be phosphorylated. So what I've shown you is this intriguing regulation in which phosphorylation of serine 114 by CDK ligase defective function have. Here is very different. So this S114 mutation, very sensitive to hydroxyurea, and we can rescue that by including the P to A variant on that background. Again, suggesting that the PIN1 activity is incredibly important for cells to be resistant to hydroxyurea. There's no replication fork start defect, and there's no increase in fork stalling. So this is very particular to the replication fork protection. It's not escaped our notice. There are lots of patient variants in these regions, many, all of which are currently unclassified clinically. So we took a look at these, and remarkably, the majority have replication fork protection defects in BRCA1 and here in BARD1. If we just focus on these ones so I don't overwhelm you with data, all of the BARD1 variants that are sensitive, all the BARD1 variants that show poor fork protection are also sensitive to hydroxyurea. The one that isn't, but they are not sensitive to olaparib, and they do not have any defects for RAD51 forming in response to irradiation. So my summary, surprisingly, the PALB2 BRCA2 canonical interaction seems to be not required for the role of BRCA1 BARD1 in replication fork protection. And instead, we have a direct RAD51 BARD1 interaction that is increased by a BRCA1 conformational change. We now have 8 previously uncharacterized BRCA1 BARD1 patient missense variants that influence fault protection, some of which have no apparent role in homologous recombination. So this was published, the majority of this was published last year, which you can read up here. We've also written a review going over some of the recent work on replication fault protection by many excellent groups. I encourage you to read that to catch up on this field. If it could be allowed a little bit of wild speculation, BRCA1 is quite remarkable. It has these three quite independent roles. It has a role in 53BP1 placement and chromatin remodeling. It has a role in recruiting PALB2 BRCA2, and it has this quite separate role, which in the context of HR is required for HR, but also separately is required for fault protection. And I think this gives us some hope. So each of these functions can be counteracted by something in therapy responses. So for example, fault protection is restored through preventing one of the nucleases being recruited, MRA11. Resection can be restored through losing 53BP1 or shielding. And recombination is often restored through hypermorph re-expression, that is a BRCA1 mutant re-expressed. But each of these, of course, potentially leaves vulnerabilities in one of the other two functions. So I think the understanding BRCA1 has these several separate roles might be useful for thinking about how to tackle therapy resistance. So only remains for me to thank my group yet again. All of these people were involved in this project in one way or the other, together with Stephanie and Xiaodong. These are our funders, and let me leave you with the important final slide.
Thank you, Joe, for such a wonderful presentation. I hope everyone learned a great deal about BRCA1 and its role in replication fault protection. We will now move on to the Q&A portion of this presentation. So please take a few minutes now to type your questions into the question box. While you are typing in your questions, I would like to just share a little bit more about GenScript's reagent antibody services. GenScript is an extremely experienced award-winning custom antibody vendor with almost 50,000 antibodies generated to date. We are able to handle a wide range of project types at different complexity levels, and our monoclonal and polyclonal antibody services are highly customizable and can be tailored to your specific research needs. In addition to proprietary technologies that can help maximize your success at generating a high-quality antibody, we also offer a wide range of specialized services like ELISA kit generation and antibody labeling. Furthermore, we are currently also welcoming researchers to test out our advanced single B-cell screening technology for reagent antibody discovery using the Beacon platform. So please visit our website or email antibody@genscript.com to learn more.